All right, we're back for one more lecture to finish up Chapter 18. And this also has an industrial connection because it's about the tests you can do. Now, chemistry instruments are very sensitive, but they're hard to take from place to place. What you need usually is you need some kind of diagnostic test that can move to different rooms in a hospital or maybe even to clinics in remote places that can tell you if you have a certain molecule in your blood, a certain kind of damage to your system. And it turns out that one of the most useful indicators of cellular damage somewhere in the body is when you see an enzyme that's not in the right place. The enzyme that they like to use for this is aminotransferases. It's a very important enzyme. They're very good at what they do, and they're very simple. Because it's an isomerization reaction, it doesn't require ATP, doesn't require cofactors. It basically, just requires the one cofactor that sticks with the enzyme, and that's pretty simple as far as things go. These are great, simple, basic enzymes, and aminotransferases, therefore, are also always inside cells. You don't have aminotransferases in the bloodstream. They're kept inside the cell. So if you see an aminotransferase in the bloodstream, you know something is wrong. There's some cell somewhere that's inside out. And that's the idea. And there's two important aminotransferases and some other molecules that are perhaps the most widespread test. Now, the one thing is, because these are named by doctors, they actually don't have the same names that biochemists use. And so you're going to have to see that it has two names for one enzyme. But if you know what the enzyme does, the names make sense. So we've talked about alanine aminotransferase. Great. And it passes from alanine to glutamate, right? Remember, that's the typical aminotransferase activity, passing things to glutamate. So you can also name it by its last name, or the products rather than the reactants. It's glutamate pyruvate transaminase. And realize that actually there's only one of the four components that's not even named here, but you can guess what it is, right? It's alpha-ketoglutarate. So aspartate aminotransferase does the same thing. Remember that we are working with the alpha carbon nitrogen, is what I'm going to say, not the side chain. But it's also called glutamate oxaloacetate transaminase because oxaloacetate is the counterpart to aspartate. So after a heart attack, you basically will have muscle cells that have a lot of the transferases in them. Remember that muscle cells process protein a lot. So muscle and liver are going to have the most transaminases in them. Blood is not going to have any. If you see transaminases in the blood, you know you've got a problem, most likely with the heart or with the liver. So the heart attack, damaged heart cells are turned inside out, and they release the GOT and GPT into the blood, or as we would call them, alanine and aspartate aminotransferases. And you can tell how many cells are broken by the amount of transferase activity that you see in the blood, the number of enzymes, if you can get it down to that. So these are called the sGOT and sGPT tests. S stands for soluble. Soluble means in the blood, in the serum. And there's actually a third protein that they look for, creatine kinase. And if you remember, creatine kinase, it was mentioned way back in chapter 14. It's in the muscle. And it actually has a specialized muscle job. But again, it's supposed to be inside the heart cells. If it's outside the heart cells, you're in trouble. The other thing about these is it's pretty easy to provide these enzymes with the substrates they want. And then you can measure their activity very easily. So the thing is, it's not just people who've had heart attacks. There's people who have liver damage, and it can be not just acute liver damage, but chronic liver damage. So they've detected high levels of GOT and GPT in chemical industry workers because they've been exposed to nasty chemicals. The liver is the place that tries to detox, but the nasty chemicals are so nasty that they lyse some liver cells, and then the contents of the liver cells are circulating in the blood. This can actually be, so the same test can test for liver damage in a chemical worker and for a heart attack in a non-chemical worker, right? So I want you to think about why they are useful in diagnoses. Why do we use these? I already mentioned some of the reasons, but maybe you can try to recall. Okay, pause it if you want to think about it. And I'm going to tell you the answer. They're abundant. You have to have a lot of them in the muscle and the liver. They should always be inside the cell. In the blood, it's a problem. And they're very active. They have a simple collection of products and reactants. They actually can go either way because they're basically isomerization reactions. And so that means that you can put in the products, what we think of as the products, and they will produce reactants for you. You put in a lot of the one thing, and it will produce some of the other thing. And so this is the kind of thing that we can do in a tube first, and that's how it was first worked out in little test tubes, right? But what if we didn't even have to have those test tubes, didn't have to do it in liquid form? Paper adsorbs liquid, right? And so you can put something on paper and it can react on the paper. Everyone's used to this now because we have all the COVID tests, right? COVID tests, pregnancy tests, etc. We're used to the idea that you can measure a chemical reaction by having a colored product produced on a paper support. So this is actually, by this time, it's a relatively old idea. This is a 13-year-old paper, but I do like the idea because it says that if you take a single drop of blood, you don't have to have anything more advanced than an eye that can see color, because you can have an indicator that will change color depending on the presence of the product of the reaction that you're looking for. And you see right here you have something designed by biochemists, because you see they have a control, and then they have an AST and an ALT dot. And they're showing you an example, I believe on the left is an example of a test where you don't have high levels of AST, ALT, or remember that that's GOT on the left and GPT on the right of that. But on the right slide of that, you have the colors. You have the blue color that's shown up. You have some AST being released into the blood. And you have a little lower level, but still definitely a color change of the ALT being released. So this is a positive result in which you have detected some kind of cellular damage by taking a blood sample. That's the great thing about the blood. It sees all these different parts of the body. And so if we can see into the blood, we can also see into those different parts of the body. Here's another paper diagnostic. This was actually made on a piece of paper towel, so it shows you don't have to have fancy paper. It's rough because it's paper towel, and they printed different dyes that would react with the different blood antigens. And so right here you have, and the readout's pretty easy. They have the design. It's kind of cool to think about how they designed this. You know, you have A positive blood in the upper left corner, and then you have O negative blood in the lower right corner. And you can see how they have an X there if you have one of the other antigens that X's out the O. You know, if you don't have any of the antigens, then everything's blank and you see the little red O. It's cool to think about how they designed these. And the thing is, we could actually do a lab like this. I've done microfluidics labs before, and I've done paper chromatography, and this is just a combination of the ideas that you have a small diagnostic test. You just need to work out the chemistry to have a colored reagent that is produced, where before you had an uncolored reagent. You put all the stuff on the paper except for the one ingredient that is missing. When that one ingredient gets on the paper, then you have a test that can be done without having a chemistry instrument that you're lugging around. This is really helpful for underprivileged areas, for areas that aren't rich enough to have a chemistry lab on hand. It allows the chemistry lab to be taken in place of paper. And clearly this allowed us to test for COVID, even in our own homes, to mail them out by the dozens to people who needed them. I think that we do medicine differently now because of chemistry. And you need to know how that chemistry works, especially if you're trying to determine the limits or the uncertainties in the tests that we have. Okay. So that is, and I will show the assigned homework just in case you want, but this is all online. Just want to remind my biochem students one more time that we have a reading that will be for test 2. All right. See you in the next class.